Okay, well, good afternoon, everyone, and welcome to this afternoon's workshop on digital collections data and tracking disease. As you'll learn shortly, this workshop has been several years in the making, and we're excited to finally have this opportunity for presentations and discussion. I'm Pam Soltis. I'm a curator at the Florida Museum of Natural History at the University of Florida, and I'm the director of research for iDigBio. And I'm introducing this workshop on behalf of my co-organizers, Deb Paul, Cody Thompson, and Courtney Fote. So many thanks to all of them as well. And I'll give you a little bit more information on each of them and their roles as I go through the introduction. So the motivation for today's workshop actually goes back 4 years and really traces to April of 2020, when a task force was organized by CTAF, which is the Consortium of European Taxonomic Facilities, and DISCO, the Distributed System of Scientific Collections. And this task force was organized to address the possible roles that natural history collections could play in addressing the COVID crisis in particular and in disease more generally. And I'll note right now as well that we had sort of an offshoot of the CTAF DISCO task force that took place here in the US that we called Virome. And some of the products of those activities I'll be showing in just a second. So the task force was led by Anna Cassino, Dimitris Kourias, and Voucher. And I'd really like to thank them yet again for their leadership, and also all of the task force members for their inspiration. This was, as I'm sure everybody remembers back, a really hard time, and having this weekly collaborative discussion of how we might be able to bring natural history collections to do some small part in coping with the COVID crisis was really valuable, I think, to those of us who are participating. And I'll note that some of the collaborations that developed during this time frame, during the task force and Virome, still continue today. And this is, in fact, a screenshot of one of our Zoom meetings, and both Deb and I were in attendance at this particular session. Now, several publications resulted from the work of the task force and its derivatives, such as the Virome project, and some of these are listed here with contributions from several of the participants in this workshop. And I'll note here that the one in the lower right was led by Cody. The one in the lower left was led by Jocelyn, and several others in the room participated as co-authors in one or more of these publications. And so this was sort of the start of us thinking about how we could continue to bring the community together. So we have so much biodiversity data and expertise relevant to understanding, potentially preventing and mitigating and addressing zoonotic and other spillover events. And we've learned a lot from past events, for example, with the hantavirus, flu variants, and of course, COVID. But our various communities of expertise rarely communicate, and they're not well integrated, they're not well connected at the data level, the analysis level, the professional level, and at the policy level. Now, there are certain members of this workshop who sort of straddle some of these fields, but in fact, the vast majority of people in these different disciplines rarely have opportunities to communicate with each other. And we'll also note that while much of biodiversity remains unknown or is threatened, that the number of humans doing this sort of work is also dwindling. And so there are multiple challenges on multiple levels. So back in 2020, Deb and I wrote a proposal to NSF to bring these communities together in a series of workshops. And this is the beginning, essentially, of where we are today. And the proposal was funded infrastructure for predicting, understanding, and mitigating zoonotic disease outbreaks. And our original plan was that we would have 2 in-person workshops in 2021, because in the summer of 2020, of course, all of this mayhem was going to go away by 2021. And so the first of these in-person workshops was scheduled to be associated with the Tadwig meeting in Gainesville in September of 21. And the second one was to be held in conjunction with the Biodiversity Summit that was going to be held in Washington, D.C. in November of 21. And like so much else during that time frame, COVID interfered, even for something that was designed to address COVID. And so both of those conferences actually went online, which meant that we couldn't actually do the in-person workshops that we were planning. So the proposal was funded, COVID continued to interfere, and the conferences then, as they went online, we decided to postpone the in-person workshops. So here we are finally implementing the workshops, both here at Digital Data and then another in June at ASM. And this is where Cody really enters the picture. So many, many thanks to Cody for his ties to the mammal community. This is something that Deb and I would not have been able to do ourselves. And we also thank Cody for his awesome organizational skills for helping to make these workshops happen. So thank you very much in advance before we get through the rest of the workshop, Cody, for your help. Now, understandably, the motivation for the proposal and given the world's experience at the time with COVID, our focus for the workshops was, as we designed them and is, bats and viruses. But this is really just the framework that we're using for addressing the larger topic, which is the need for better infrastructure for including communication and collaboration, but better infrastructure for predicting, understanding, and mitigating disease. So most of the presentations are going to focus on bats and pathogens, but our discussions are not restricted to bats, nor even to mammals, and neither are they restricted to viruses. So as we get into the discussion part of the workshop this afternoon, we're happy to think about how to extend out to other organisms, other types of disease, and other sorts of vectors. So we recognize the need for action for gathering, sharing, and transferring data. And so our workshops are really focused on building fundamental bridges between natural history collections and all of these things that are listed here in order to establish a dialogue and to provide a framework for integrated research and for an integrated research agenda. So as we attempt to learn from each other about understanding these processes, we all need to communicate, and there are multiple ways that we can be effective at communicating, and we need to communicate this knowledge not just among ourselves, but to other stakeholders. And this is where alternative methods of communication come into play. So through live graphic recording, we can gain a different means of communication, and this is a path toward conversations and maybe post-conference events that can help us convey what's happened here and also what maybe the next steps will be. And so this is where we'd like to welcome Courtney Fote, who's the advisor for strategic initiatives and organizational engagement from the Watson Library here at KU. And Courtney is joining us to do three things. So first of all, illustrate the talks of our speakers. We have an example here. Also to provide a graphic analysis of, or graphic synthesis of the ideas and the lessons being shared. And then also to give us an example of the value added to our own scientific activities when we include the power of the arts in our communication strategies. So this is an example of the work that Courtney will produce. This is a draft of the graphic summary from Kelly Spears's talk yesterday. And isn't this just an amazing representation? So Courtney will develop a similar summary for each of the presentations this afternoon, and then she will continue to capture the essence of our discussion session. So here is our workshop overview, and there are a few changes to note. As we heard this morning, Jocelyn Colella is unable to join us today, so she will not be present, so we'll just take a break at that point in order to be able to keep on schedule. because there is a posted schedule and there may be people who are planning to jump back and forth between sessions. We can use that time for some discussion if there is any, but we're going to take a break there. And then a little bit later, we'll focus on some case studies. And in this particular session, Neal Platt has been stranded in Phoenix for 2 days, even though he was coming from Charlotte. And he will not be able to make it here in person, but he'll be presenting remotely. So our speakers are going to cover a range of topics in these, address a number of questions that we asked in the actual grant proposal. And you can see a list here of some of these topics. And then we have some deliverables that we promised to NSF as part of this process. Now, though, because this is a workshop and not a symposium, there are assignments for all of you as well. So don't automatically head for the door when you hear that. But we all have roles to play because, in fact, this is a workshop and we're interested in gathering perspectives and insights from everyone who's here. So we ask each of you to go to this Google Doc and add your name and contribute to the notes for the workshop. And what we'll also ask that you do is to please identify one key point to share during the discussion. These questions are noted in the Google Doc, so you can think about them in advance. You can jot your own personal ideas down, either anonymously or with your own name associated with them. But these are the questions that we'll be focusing on a little bit later. So our grant deliverables are listed here: an improved vision, greater understanding of the role and need for biological infrastructure in connecting biodiversity to human health, opportunities for public and scientific engagement, and the recorded lectures and any other educational materials. Those are the deliverables that we're hoping to get at through this workshop and the one at ASM. The ones that are not in bold: a common language for ongoing discussion and a conceptual framework for integrated research, are things that we're going to be hoping that we can generate through synthesis and discussion. Another possible deliverable is that bioscience has a call for papers on One Health, and so we'd like to contribute to this effort as a product of the workshop. So if you're interested in contributing to this, we'll be reaching out to those who are interested, so you can indicate that on the Google Doc as well. And authorship will be available to everyone who participates. And also, we hope to be able to share Courtney's collection of graphic recordings across multiple venues as things go along. So finally, just for a few acknowledgements, we'd like to thank the Digital Data 8 organizers. So Andy, thank you. Jill and the rest of the IDIG Bio staff, thank you for all of your great organization. To our speakers in advance, thanks for sharing your expertise. And to all of you, our participants, thank you for sharing your thoughts and insights. And finally, thanks to NSF for funding this 4 years ago. So I think that's it for now, and I think we'll go ahead and move on to our next talk, and that will be given by Cody Thompson. Let's see. So we'll just, yeah, do we want to, okay. How much we're going to try to, I guess here you can't.
We have a 15-minute buffer, right?
Yeah.
I'll try not to use it all.
A quick reminder that the talks are scheduled to be 10 minutes with 5 for Q&A, but we have a little bit of a buffer, so.
Okay, thank you, Pam. Yeah, so my name is Cody Thompson. I'm the Mammal Collection Manager and Associate Research Scientist at the University of Michigan Museum of Zoology and the Department of Ecology and Evolutionary Biology. So Pam kind of alluded to it already, but my talk will be centered around this idea that we put together in 2020 about really pushing to get museum collections involved in pandemic preparedness, host pathogen research, et cetera. It's something that has been ongoing for quite some time, but kind of under the radar. So really kind of moving collections to the forefront. So Kendra, who will be speaking here later, she and I led this paper that was published in Bio that will be kind of the basis of this talk. So first of all, remember this. Hopefully this image isn't burned in all of our heads. Sometimes I wonder, listening to the news, whether or not it's truly been burned into everybody's heads. But certainly this image and this virus changed the world over the past 4 years, and I think we all can acknowledge that. We don't want that to happen again, right? But as we all know, there are things that are in the background. So avian influenza is one that's currently in front of mind that's moving around, but certainly we do not want to be on the nightly news looking at images of pathogen structures. Another thing that we all should remember the past couple of years are statistics like this. So this is from the Johns Hopkins University website that was focused on keeping a relatively good tally of cases of SARS-CoV-2 or COVID-19, the deaths that occurred, in addition to the vaccines being administered once that was possible. So if you look at, this is an older slide, but if you look at the date up there, this is from December 2nd, 2021, so a quarter of a billion cases were documented. So we can all know that that's probably a drastic underestimate. And I'm sure if we were to find these numbers today, we'd be talking about numbers in the billions in terms of number of cases. And the deaths would be certainly much higher than what we're at the time. But part of that, of course, is that last number, is that we were reaching a point where vaccines were being administered, people were that had been infected had developed some resistance. So we're kind of getting to a point where things were a little bit starting to calm down, but we definitely were not over the hump, right? But one thing that's interesting about SARS-CoV-2, and Kendra probably remembers this slide, is that it has connections to the flora and fauna of the planet, and that's where we come in as museum scientists. And this is our expertise. This is what things that we are passionate about. And if you look at that number, 60% of all human infectious diseases have a zoonotic origin. That's a pretty impressive number. But again, I think that kind of gets lost as those information gets moved around in our modern news and whatnot. But I think our part, of course, is connecting those connections to people and making connections to our environment. And just seeing those images of those animals in the corner, I think that does hopefully bring connections to those of us that are mammalogists, for example. We study those type of organisms, so we want to know how they're interconnected with our everyday lives. Of those 60% of viruses, 75% of those diseases have originated in wildlife species, and then there's some good examples here. We often think about bats, and maybe bats sometimes maybe get a little bit of a bad rap, but they are certainly involved in a lot of the zoonotic outbreaks that we see. But you also have things in there such as deer mice, an animal that you can go out 100 yards away from this building and probably find. We have chimpanzees, we have mosquitoes, ducks, camels. All these things are integrated into potential disease cycles. And why is this happening? I think this is an apparent part of the process that we all can appreciate here as we think about how the planet is changing and how we go about documenting that change in our own work, whether it's looking at habitat fragmentation, the effects of climate change on species distributions. We know that that's happening. And as a result of that, we do see these dramatic changes in the pathogen environment that leads to spillover events. And these are just some of the more notable cases that have happened recently. And one might argue, well, maybe that's a product of our ability to survey better now, but it most certainly is related to the changing environment that we are now rapidly seeing in the past century. So Rick will probably argue with me on this particular slide, but all human coronaviruses have many other things involved, right? They don't just, at least as far as we know, again, Rick may correct me on this, they're not jumping directly into people, right? There's a lot of cases in intermediate host. For example, MERS, the camel picture that you saw below, is definitely involved in that transmission cycle. But you notice there's a lot of question marks here, right? I mean, there's a lot of places that we don't really have a good appreciation for, and especially as it relates to SARS-CoV-2, I think we are all acutely aware of a lot of the different theories that are out there that relate back to why that particular virus made its way into humans. I must have saved the transitions, but sorry about that. But one thing is for sure is the evidence could have been there, right? And that's where the paper that Kendra and I were involved with and led, some folks in here that were involved with led or participated in, we do know, right, that there was sequence data that was published in GenBank that had data that was pretty similar to SARS-CoV-2, but you would be hard pressed to figure out where they actually came from, what animal host, whether it's an intermediate host or primary host, that data connects to, because oftentimes the host just says bat, right? And bats are the second most populous order of mammals in the world, and so there's quite a few bat options to choose from if you're just putting down bat. So this particular paper, we focused on that. We focused on, why is that the case, and how do we need to change that? Okay, so we make this case for vouchering. Moving along here, so this is, again, I think where museums come in. I think if anybody knows about Joseph Grinnell, I think a lot of us, our collections are connected very intimately with the work of Joseph Grinnell and his push to really have a systematic way of collecting data and preserving specimens over time. And Grinnell, I think a lot of mammalogists, we claim him as ours, ornithologists claim him as theirs as well. But Grinnell really was instrumental in making the data that we now take for granted oftentimes, making sure that it was here a century later. So he was really active in the 1910s, 1920s. He had this really prophetic statement that essentially says, we're doing things now, right, for the future. I'm going to go out and collect this material now for this particular project, but I'm going to do everything I can to make sure the data, that the specimens are preserved are good, that the specimens are preserved in a way that they could be used for research that I don't even understand. And of course, in 1910, he didn't even know what the structure of DNA looked like, right? So he was really thinking outside of the box at the time. And of course, for those of us that, again, are tied into this world of getting our data out, this is the result of all that, right? I mean, a lot of this, of the 3 billion records that are currently on GBIF, a lot of those, not the vast majority, because you have things like iNaturalist and eBird pumping data into there, but a lot of those go back to museum records, and integrating those with the citizen science efforts that, like I mentioned, iNaturalist and eBird and others, you have a really, really valuable data set that, to be honest, most of our colleagues in public health have no idea even exist. Most of them have no idea that they could get samples from our collections, let alone go to this freely available data site and download data on a potential host of the viruses they study. So, what I think that Pam kind of set the tone. What you'll see here for the rest of the talks is that really collections themselves are really kind of almost a baseline infrastructure that needs to be in place for really doing long-term predictive modeling, long-term surveillance work on these pathogens. And if we can go back to these drawers and find these mice or birds or whatever it may be, and connect those back to the tissue samples, we have a really powerful mechanism to do the research that is necessary for preventing something like COVID-19 from happening again. And this is a slide that I think I might have stole from Joe Cook at one point in time, but I think for us to do that, the data, which is going to be a big thing that we talk about today, but also the specimens and the way we collect. We can't be focused just on the current research question of the day, right? We have to be thinking about what the tomorrow questions may be and do everything we can to get those data and those samples in front of people that are interested in this space. So, really being diligent about producing holistic specimens and holistic data approaches. And I think what we need to do at the end of the day is trying to merge these two different communities, right? And I think that's the whole goal of this workshop, is to try to get different folks to talk a little bit more than what we currently do. But and that top slide is a good example. We have people that are going out and surveying bats for pathogens, but those bats oftentimes end up incinerated. Good example are most state rabies testing labs. Those bats oftentimes are incinerated, and oftentimes are not correctly ID'd after they may have rabies or other things in them as well. But we want something more like this, right? We want these fluid preserved collections, dried collections we have, the complementary tissue samples that go along with them, and make those available to these communities that are doing the work. So I guess the question I'll leave to the group as we kind of head into the next set of talks is what do you believe the natural history, what can natural history collections do to be part of the solution? I think we often at university we kind of get this ivory tower mentality and don't really think always about the applied nature of our work, right? And that's, museums, we're not immune to that. And I think that's something that we need to think about more, how our work, our research, our collections relate back to the public, and really to truly justify our existence, we have to be doing that, and we have to be doing that in many ways as possible. And this is one way that definitely has a profound effect on the global community. Okay. All right. Thank you. So I think we have a little bit of break here, but we have some time for questions before we move on to the next talk. Rick. Wait.
Well, excellent talk. I absolutely firmly disagree with you on the Corona stuff, but I'll get to you on my
Talk about
talk
that later.
I'll tell you later. So I think you put something there really great, which is something we've struggled with, even in data science when it comes to like databases and stuff like that. How do we get the funders' mindset that yes, this is a question we need to answer, this is a research topic, but please understand that when we take it for this one topic, for this one question, then in our grants, that this is also a resource that has a longer term investment on, and then getting their mind away from this kind of short 2-3 year mindset to a more long lasting. And again, I don't expect anyone to have an answer for this, but how do we get their mind off more long term? Because in physics, they get 10-20 years to solve a question. Why can't we get that in biology? So I just curious people's thoughts on this.
Yeah, I'll take a stab at it.
Yeah.
I think for the museum world, when we hear about colleagues that are getting grants and they want the deposit material in our collections, oftentimes we say, well, how much money is coming to the collections, right? And I know there are people, Andy and Pam and others, that have been heavily involved in making sure that longevity exists beyond the 3 or 4 or 5 years of the grant proposal, right? For museum collections in our data, talk about a perpetual problem that I don't think we fully have gotten a grasp on, because the infrastructure costs aren't getting cheaper, right? The servers to put the data out there are not getting cheaper. And I think you're right, though. That's thinking about the project itself, which probably does need more time in a lot of cases, but those data should live forever, right? And I think that's the difficult part. I think we're getting better at it, though. I mean, I think efforts, but there's, oftentimes we get, again, kind of caught in our ivory tower mentality, the protective of our data. We want to get data out. We want to make sure it gets to the right people. I think that's the other thing that's always hard to understand. But in this case, public health data, we want to get it out quickly, right? We want it to get into decision makers to make decisions on stuff that needs to happen fast. And also get tenure and promoted and all that sort of stuff goes along with it.
I understand that, and I completely agree, but just getting their mindset around that things take time. We need longer times of money, not a 2 to 3 year turnaround on some of these things, and that the impact will be exponentially larger we've had Nobel Prize laureates outright say that we just need more time, and being able to turn around what they want with more and bigger folks and bigger, more multidisciplinary, but yet only a 2 or 3 year project. It's just impossible. Right. And just getting their head about why can't they think of a long-term vision? And I don't care what agency that wants to have this vision, but we all need to come together as a community and say, look, we need a 10-year approach, 20-year approach, not a kind of flash in the pan, 2-year kind of grant, right? And I don't think we're going to really have they say, where's all this translation? Where's all this stuff? And how do you turn this stuff around in 2 to 3 years? I mean, it's hard to even get people hired, especially during COVID, in 2 years, right? To turn these things around, right? Mm-hmm. And so we just have to come to a community and say, look, we need this to really do this well.
Right. And I think for the surveillance stuff, right, getting permits, as I've been educating you in the past, in itself could be, it's an ordeal, right? So
just this whole short frame mindset, we just have to get that around.
And I think that was one of the fundamental premises behind the Biological Collections Action Center, is that this would be a long-term funded entity that would be able to coordinate this kind of stuff and wouldn't be this stochastic 2 to 3 year funding cycle. It would be a much longer funding cycle that would be able to perpetuate these things and keep them going.
Right. Somebody else had their hand up over here. No.
You don't want to talk, Deb? Will.
Yeah, I'll call on people. Just wait. I would add to that, I had the pleasure and privilege of being part of an NSF webinar where the talk was about the bioeconomy. And I don't know about how many of you get to have conversations with economists. Anybody here? You? Yay. Thank you. So I had the pleasure, and the economist pointed out something. I knew that the data is valuable long-term that we collect, but I didn't have the economist's perspective. And in this case, she said that our data is quite unusual. From an economist's perspective, when they're trying to calculate value and understanding that what the data can do for them, that our data actually grows in value over time because of the way it provides baseline information, because of the way it provides grounded items that people can go back and look at and touch and refer to. It's a data set like no other. And we probably don't really grasp that value enough in terms of presenting that to people who are going to fund what we do. And it gets to this point that you're making. There's an NIH paper. If any of you can find it, I can't find it anymore. It's a white paper that about, 6 or 7 NIH researchers wrote. And they were echoing what you're saying, Rick, about the 5 and 10 year funding cycles where you're just getting going and just starting to understand something when everything just basically goes out from under you. And this notion, if these kinds of things we need to do take a longer window to address.
Yeah. Pam?
If you want groundbreaking stuff, it's got to be done.
Okay. Here.
Sorry. So I think if you're going to have really groundbreaking stuff that just is that is truly translational, you got to have more time, right? And I just want to hammer this into the minds of program officers that even if you're already set up to do that thing, a 2 to 3 year thing, it's just not gonna work over in the long term thing. And I just think we have to sorry.
Thanks for that point. Hopefully somebody's listening. So I wanted to get back to the point that you were making before about vouchers, Cody, and and the reason, and this also ties into the value of the data, because when the pathogen data are connected to the vouchers, then that provides that sort of baseline information that we can eventually go back to in the future. One of the activities that we participated in through that CTEF DISCO task force was developing a new portion of the database for GenBank, so that GenBank submissions of COVID data actually had very easy access to providing voucher information. GenBank couldn't require that the voucher information be made available, but we, with GenBank's help, were able to develop a new sort of entry form for that. And I haven't gone back to see if that actually helped to facilitate adding more voucher information, but you know, there are opportunities to potentially make some make some contributions from our community that could help with this picture. And maybe now would be a good time to actually revisit that when they're not totally swamped with all of the sorts of activities they were working on, day and night, just getting the data in. So I don't know if that's something that you guys have followed up on. Do you know if there's been any additional vouchering since, the paper that you and Kendra led was awesome. Do you think that's having an impact?
There has been additional vouchering, but I think actually it's gotten a lot harder for those of us that work with mammals and birds. A lot of it because of regulatory reasons that have been added since COVID, and some of it's necessary. I think we all can admit that there's things that we probably could do better and be more proactive to monitor what's coming in and out of the U.S. or any other country for that matter. But I think sometimes the red tape gets in the way of making legitimate progress on answering a really crucial question, and it doesn't make sense in a lot of ways, right? What we're doing for international collections coming in is not applied within the country. Sometimes within some states they're overly even more restrictive than some of the federal agencies are, and it's just, I think it's, and again, not that there's an intentional effort to limit the work that we do. I just think the reaction maybe went farther than what was necessary, and for us, I think that the reality is we need to get more specimens, more samples, more connections to virologists, pathobiologists, and others that don't typically come to meetings like this, and because they're the ones that have at least the technical expertise to do that type of work, but they also don't have the technical expertise that we have to do the field work, know the organisms that we're interacting with, but also ensuring that those specimens can be utilized for something entirely different later on. So I think actually we probably should move on to Kendra here. So I'll introduce Kendra, I guess, real quick. So Kendra Phelps is from the EcoHealth Alliance. Kendra will be talking about a harmonized taxonomic resource and is critical for accurately interpreting host pathogen interactions. Kendra?
Lower this a little bit though. So good afternoon, everyone, and thank you, Cody, for that introduction. So today I'm going to try to cover 3 different new resources that we've created to provide researchers such as yourself with greater access to published literature, particularly on ecological interaction data such as host pathogen interactions, while also ensuring that there is consistency in taxonomic alignments. And while I'm listed as the presenting author, I'm actually just one member of a very diverse team of international researchers who have expertise in taxonomy, disease ecology, data extraction and management, and of course, a deep appreciation for bats. And this team was brought together in 2020 as an initiative of the CTAF DISCO COVID-19 Task Force, which Pam did a great job of introducing. I initially was on a different team with Cody, and then once that disbanded, I think in 2021, I was scooped up by this other group to join, and we refer to ourselves as the Be Happy Working Group. And our initial goal was first to build workflows so we could extract published data on host pathogen interactions. And that was particularly focused on bat species that were reported to be hosts for potential pathogenic coronaviruses. But as outlined in the viewpoint published in the Lancet by some of the members of the team, our ability to achieve our end goal, which is on the far right, of interpreting host pathogen interactions was met with some significant challenges that these three resources that we developed try to solve. So the first challenge being that a lot of the relevant ecological interaction data is quite difficult to access. And this is because the data is either not in digital format, still it may be because of age, if you think about papers from the 1800s, are still only in printed format. And then we also have a lot of data that is restricted due to paywalls. And I think it's estimated like 70% of currently published scientific literature is behind a paywall. So a significant challenge. Another challenge common in data mining projects like ours was this constantly evolving knowledge of bat taxonomy. So here's just a very simple example of species A is split into two distinct species, but yet currently there is no reproducible way in which we can track such changes in taxonomy, and this leads to inconsistencies across published data sets or different databases that are out there. And in the case of host pathogen interactions, this may lead to misinterpretation of routes of transmission risk. So first, to overcome the initial set of challenges of liberating dark data, we focused on building a repository of bat-related literature. And if you scan that QR code, I don't know if people can do that on Zoom or not, you will go to the GitHub, currently it's on GitHub repository that houses, oh gosh, I don't even know how many points of literature at this point. But we use this wealth of open access literature, and we trialed some different data mining techniques to build networks of eco interactions. Today I'm just going to discuss co-roosting interactions, which is biologically relevant when considering potential routes of virus transmission between different bat species. So the bat literature repository vastly improves our accessibility of scientific literature on bats, including previously inaccessible dark data. And while we are continuously adding to the literature in this repository, it does contain top-tier content such as 3,500 journal articles and books, as well as more than 7,000 supplemental documents and notes. Concurrently with building this literature repository, we were also mining it ourselves for co-roosting interactions, since this is a type of behavior that provides ideal situations for viruses to move between individuals either of the same species or among other co-roosting species. And also other organisms that may be exposed to bat excrement. So what we have depicted here is, for example, if a bat sneezes in a cave, who would be on the receiving end of any aerosolized viral particles, whether it be a bat of the same species, a bat of a different species, a human like a spelunker in a cave, crickets, salamanders, and those types of ecological interactions are quite interesting. And in this way, we can begin to construct a network of potential pathogen transmission routes. So in regards to the co-roosting project, we've compiled over 17,000 records of co-roosting interactions, and this covers nearly half of all described bat species. And at first we were really excited to see our first, you're not meant to be able to, I thought never in my life would I show an image that I'd be like, you can't read this, sorry, I apologize. But it's just to depict that this is our first network analysis of bat co-roosting data that was mined from published literature that spanned 160 years. So this is including some of that dark data that's hard to access. But when we looked a little bit closer at the individual species nodes, which I'll show an example later with zoomed in to, we realized that taxonomy was going to be a significant challenge. Because as you would expect, taxonomic alignments over those 160 years have evolved quite a bit. So to tackle the challenge of taxonomic inconsistencies, and I might argue that this is probably the biggest challenge that we've attempted to tackle, we developed a tool to continuously track changes in taxonomic assignments. And this is to ensure accurate phylogenetic associations among bat species. So I'm going to very briefly review this, but to ensure that we're using up-to-date taxonomic assignments in our data set, the team has collected and assigned species treatments across several primary taxonomic authorities for bats, which are all 5 listed across the top. And this led to the development of the bat taxonomy alignment tool, which we call BTA for short. And it's a dynamic and versioned name translation tool. So the BTA provides a standardized way of aligning and comparing taxonomic treatments across time so that you can identify any inconsistencies in your data set while also maintaining data provenance. This approach aligns with kind of the blockchain method that was presented by Dr. Peterson yesterday morning. But ours is generally in a large spreadsheet, not in blockchain. So how different are these taxonomic alignments from one another? Well, the numbers listed in the table represent the total number of unharmonized, so that means inconsistent names, between each of the different taxonomic authorities. And results revealed that about 15% of the 1,500 binomial strings, or species names, do not match across these widely used authorities. And so clearly there are implications on interpreting interaction data sets when they're based on which taxonomic authority you're following. So when the Handbook of Mammals came out in 2019, that was a significant leap in taxonomic advancements and changes from the previous mammals species of the world published in 2005. Unfortunately, the Handbook of Mammals is already outdated, even though it was just published 5 years ago. So there's been a real push by the Mammal Diversity Database in conjunction with batnames.org. Both are spearheaded by members of our team. And they have done a great job of updating and will continuously be updating these dynamic sources of taxonomic authorities. So when we took the BTA tool and applied it to our own co-roosting network, we identified that out of those 633 bat species in our data set, 139 had inconsistent taxonomic assignments across those authorities. So that's a significant proportion. So for example, I've zoomed into a section of the network analysis that on your left includes two previously distinct species of Tidarida. So we have Tidarida brasiliensis and Tidarida cynocephala, which are both highlighted in green boxes. More recently, though, Tidarida cynocephala was designated as a subspecies of Tidarida brasiliensis. So these interactions had to be reassigned so that cynocephala, any interactions that species were having with cynocephala, now were contributed to Tidarida brasiliensis, which when you look at the two panels, you can see there's a significant difference in the structure, but also the strength of relationships between Tidarida brasiliensis and other bat species. So I moved through those resources really quickly. I should have mentioned each one of them have a QR code that leads you to a website, either on Zenodo or GitHub, that can be searched for much more details than I could provide in 10 minutes. But collectively, we wanted to use these resources for many different applications, and we think there's many different types of users that would find them very beneficial. But most importantly, ensuring that valid scientific names are easily traceable, but also the ecological information that's included when you update these species treatments. This will help with improving our ability to actually interpret host-pathogen interactions. And with that, I will answer as many questions as possible. Yeah.
Thanks, Kendra. Great talk. I have two kind of small questions. The first was, did you find any cool patterns in the co-roosting interactions that might have insight into the viral transmission in particular from doing it this way with this network analysis and the dark data? And then the second was, how do we set it up? And I think you mentioned like relational databases, but such that when the next taxonomic compendium comes out, that you can continue tracking these changes with taxonomy as it changes through time.
Yeah, so, well, to answer your second question, I think the mammal diversity database will be the go-to for that. And you know Nate Upham, who's leading that, and I think he has a small army of students that are data scientists that will continuously be scraping new data as species are described or changes in taxonomic assignments happen. Your first question about any interesting co-roosting interactions. Honestly, I have to say it's been a few months since we looked at that, but from my own research in Western Asia, we have really noticed some interesting patterns of species of different genera sharing the same virus. And these typically are co-roosting in caves. And I think caves are probably the highest, if you pick a habitat type, where you're going to have the greatest risk of transmission between host species or human interface. Caves are a very interesting interface that we should be looking at. But the fact that species that, like a Rhinolophus and a Myotis are sharing the same exact virus, and typically it's in the same cave, but not always. So that's where you start pulling in all these data sets and then adding on viral information to them, along with co-roosting patterns that you can, instead of just having this bipartite network of virus bat species, you're adding in all these other ecological interactions, which I think makes it more biologically relevant when trying to determine spillover risk. So it was a long answer to your short question.
How often is the BAT taxonomic tool updated?
So quite frequently. at least multiple times a year. Typically publications as soon as they are released, because now things are a little bit, dealing with the dark data going back to the 1800s was very difficult to pull out that kind of information. But now, especially with batnames.org, a lot of researchers submit their changes in taxonomy directly to that website, but we always have alerts for anything new that is either described or changes in taxonomic alignment. So it is still a hefty load at this point, but I have great faith in our data scientists that they will be able to automate a system that will continuously, without so much manpower, be able to keep it updated.
Okay, thank you, Kendra. Sure. Yeah. So for all the speakers, there are actually additional questions in the Google document. So feel free to hop on there and interact with online folks. And there it is. Okay, our next speaker is Verity Mathis, who will be talking about Virome composition in fresh bat guano, frozen bats, and fluid preserved bats. And I'll point out that these next set of talks are more like case studies that have taken museum collections and done or utilized them actively in pathogen based research.
Thank you, Cody. Even though I'm here presenting this data today, this data set truly belongs to my co-authors, Carla Mavian and McLean Bassett, who weren't able to be here today. So hopefully I'll be able to talk eloquently about the research that they did. I'm mostly presenting this on their behalf. My role in here was more as from the museum side as the collection manager, helping facilitate access to the collections. So as we've probably already heard multiple times today, about 60% of known pathogens are zoonotic, with about 75% responsible for emerging outbreaks. And changes in land use and climate change can influence and increase the risk of the zoonotic pathogens crossing over into human populations and increasing that spillover risk. And so natural history collections really, like we've emphasized a lot today, stand at the forefront as they are spatial temporal distribution maps of biodiversity. And so we can use these collections to help answer questions about spillover risk by tracing the spread and identifying origins. And the question is, how can we best utilize these historical collections to do those things? So I'm a mammologist, so I'm going to be throwing in the cute bat pictures whenever I can. So we are using bats in this case study. And in about late 2020, Carla Mavian approached me. She's in the Emerging Pathogens Institute at UF. And well, she first approached Pam and then I think me about using the natural history collections to kind of look at the coronavirus. And bats, of course, were the natural method to do that at the time. So Florida is home to 13 species of bats, including the big brown bat, Eptesicus fuscus, which is a known reservoir for coronaviruses. And coronaviruses had previously been found in Florida. And bats do harbor a significantly higher proportion of zoonotic viruses than other mammalian orders, and they're a known reservoir for a variety of pathogens. So they were a very ideal resource to use for this study. We kind of got this seed grant together to kind of use the collections to answer these questions about whether or not we could use the collections to predict the risk of bat populations to be reservoirs of coronavirus or other pathogens by analyzing the gut microbiome. And so our goal was to identify the biomarkers that could be used to predict the host suitability. So we drafted this pilot study to kind of demonstrate a proof of concept and see if we could determine the feasibility of answering these questions using a variety of preservation types. And so the Florida Museum, we have over 2,600 bat specimens that are represented in some shape or form as a fluid preservation. Some of those are full whole body fluid preserved specimens. We have GI tracts in fluid, we have carcasses, we even have skins in fluid of bats. So we have a pretty good resource to answer these kinds of questions. And so we focused our study on 4 species of bats that are very common in Florida. We have the Brazilian free-tail bat, the evening bat, the southeastern myotis, and the Seminole bat. And we chose these because they were really well represented in our collection as both formalin-fixed specimens, as ethanol-fixed, and for a few of these we had a frozen backlog that we could utilize as well. So we, because this is a project pilot study, we kind of kept the sample size to a relatively manageable amount because we wanted to see if the methods would work out or not. So we tried to aim for about 5 from each of these preservation types for each of these species. We were able to do that for most of them. As you can see, some of them we only hit 4 for that preservation type. And then also, along with the museum specimens, Carla was able to obtain about 14 fecal and oral swabs from Tadaridae brasiliensis, working with our local Fish and Wildlife government section who are out netting bats. So altogether, we had about 36 specimens that we were able to use for this project. And we didn't have any frozen bats of Leserius or the Nycticius, the evening bat or the Seminole bat. So only the free-tail bat and the southeastern myotis are represented in that aspect. And the age of our museum samples range from the early 1930s to 1960s for the formalin and the ethanol. And then 2021 is the frozen specimens that we had in a freezer. So this is a representation of the overall workflow that Carla's lab did for the 2 different sample types, the fecal swabs on the left and from the fresh samples, and then on the right being the museum samples. And it's basically just a workflow showing the various methods that they use to get the RNA and the DNA. And then they, for the RNA, it was reverse transcribed into the cDNA, and they used Illumina sequencing to do shotgun sequencing. And if you have any questions about the details of the methods, I can definitely get you in touch with Carla about that. So this is just a breakdown of what kind of worked and got results for the DNA and the RNA for the different preservation types. For the museum samples and then for the fresh samples. And you can tell we really didn't have too much luck with the museum fluid-based preservations. We had some luck with the myotis and the Tadaridae, but really nothing for, very limited for the Nycticius and nothing for the formalin really, except for some of the Tadaridae. So kind of disappointing, but we'll continue on with the results. So this is a heat map representing the blast results of the RNA contigs that were generated and stratified by the input sample type and the virus classification. And the color is on the heat map is the length of the match identified by blast. So the proportion of the contig that overlapped with the blast that was the blast hit from doing the searches. And it is interesting to note here is that she was able to identify bat coronavirus and bat astrovirus from the frozen myotis samples. And then also endogenous retrovirus from Desmodus rotundus, which is the vampire bat, was obtained from one of the fecal swabs from the free-tail bat. And this is just 2 maximum likelihood trees of the contigs that were identified corresponding to the alpha coronavirus and then the astroviruses compared with the most closely related sequences from the NCBI database. And so with the red boxes indicating the frozen myotis samples that had amplified and matched up, and kind of it is worth noting that the color arrangement is not a one-to-one match here between those two figures. So you kind of have to look at each legend separately in respect to that. But so you can see that, the myotis, I don't know how well you can read that, probably not too well, but mostly the myotis is related to other myotis and pipistrelle species from North America and Europe, I think. And it is worth noting that I did want to point out that the frozen myotis that yield these results came from a colony that were living in a fire station, so within pretty close proximity to humans. So we did know the origin of those guys. And this is just the same one for the endogenous retrovirus tree, showing from the fecal swabs that were obtained from the free-tail bats in relation to the most closely related sequences that were identified by BLAST. And these were from the live caught bats that were caught outside Camp Blanding in Florida. And finally, these are the gut microbiome results. So using showing the bar plots of the metagenomic DNA sequences breaking down by the metaplan classification, which is the metagenomic phylogenetic analysis. And so it takes the input from the raw reads and then doing some basic quality filtering and trimming. But it is worth noting that the samples that represent a minority species or fell below a certain cutoff were classified as other. So if you look at the myotis, the frozen myotis species there, they're pretty much all like others. So we really weren't able to identify any true kind of information from those guys. And then the rest were the fecal swabs. And I think one of the Nycticias humoralis yielded some results. So it's kind of a presentation of negative results almost, in that we ultimately did not have too much luck with the formalin and ethanol preserved historical specimens. There's always, the age of the samples might have played into that. You always have to take a guess at the original preservation methods. You really don't know, especially for those older 1930 specimens, how they got into the fluid and the history that led up to them previously before getting fixed. But we were able to identify potential coronaviruses and astroviruses from the frozen bat samples. So this kind of just leads to the questions that I have for this workshop. And as a museum professional who's in charge of managing collections, what can we do better to ensure better preservation and variety of preservation types for future microbiome and pathogen research? And then also, how do we best database and link these kinds of data to the voucher material? So we have these results, but how can we link them back to the voucher in a really easily obtainable way that can make sense to others who are trying to find it? And with that, I think I have time for questions.
Time for one or two questions for Verity.
Oh boy, I'm in trouble now.
Can I just reemphasize again that I'm a mammologist and not a microbiologist?
All right. So I wanted to just thank you because you've led beautifully into what I'm going to talk about in a minute. But I also want to say that, again, I think I have a solution for all this stuff that you were talking about. But I'm just fascinated by trying to get at some of these other collections, and in that, how many frozen bats do you have, and how are you keeping those frozen? And then that's a pivot point, which I think we should definitely talk about. So what are your thoughts on that?
So in terms of numbers of frozen bats, it varies. We try not to keep our backlog sitting in backlog for too terribly long, although I will admit I still think some of those 2021 bats are in the freezer at this point. But a lot of that is just time and taking
So they kept it like minus 80?
Minus 20. Sorry.
Okay.
So they're in a minus 20 test freezer, which is not ideal, probably.
Yeah.
Especially since our test freezer is, ancient. But so they're generally kept in negative 20s, and then prepped as soon as possible. But If always just
That's so unfortunate, right? Because, if they're -80 or flash frozen, then we can get at the whole genomes. But again, this will bleed very
But we do
beautifully.
have a -80, so it is worth maybe potentially trying to find the space for certain
Yeah.
I mean, the space is limited in -80, of course. But
There was an interesting paper that was published out of Hong Kong, and they were trying to look at like what tissues to preserve for various pathogens, and I can maybe send that around.
yeah.
Obviously for coronas and these types of things, obviously lung, but even something like stool is a place to look. And there's a lot of these other preservatives that I just like that we should just have a conversation about preservatives, because I think this is, I want to be able to study these things from the 1970s, or, even older up to now, because I can
Mhm.
really reconstruct what's going on. But
Yeah, like knowing like the best tissues to preserve and if they need to be, buffered, unbuffered, one of
mean,
each, that kind of thing.
we
Like
have
kind
struggled
of
with this. I'm not going to talk about it today, but we have struggled with this immensely. I think I have a solution, but
excited to hear it. Yeah.
Straight into liquid nitrogen.
Yeah, that would be ideal for sure.
And it's methodological.
Not molecular. I'm curious with the bats that you classified as formalin and ethanol from the 30s. Did you have good data that indicated how they were preserved? Because I think we're terrible at doing that as mammalogists.
And I'm hesitant to say that. I don't know how much to trust a database, right? So we don't necessarily have a strong history of documenting those kinds of things. So I was just kind of trusting the database, saying if it was formalin fixed versus ethanol. But that is a very good question, and that, considering neither of those really yielded much in the way of data, I don't know if that really mattered too much, but I think that is a very valid question, and we need to do better at tracking the preservation. And we have gotten better about that, but those old historical samples are a little bit of a black box mystery sometimes.
cool. Thanks.
Thank you, Verity. I'm kind of impressed that Verity only has specimens going back to 2021 in her freezers. So next, the rest of us, that looks so good. Okay, our next presenter is Molly McDonough and Adam Ferguson. Molly will be talking about exploring bat coronaviruses using the Field Museum of Natural History cryo collection.
Thanks, Cody and Pam and Deb, for inviting me and giving me the opportunity to talk about this experience Adam and I had working with the Field Museum's cryo collection. And I'm definitely coming at it from a user side. So I'm a faculty member at Chicago State University and research associate at the Field Museum. And so I'm working with the specimens and also these tissue samples. And Adam is a collection manager of mammals at the Field Museum. So he's coming at it from the collection side. This is a project that we initiated during the pandemic when we were still working from home. I was teaching from home on Zoom, and the students were not turning on their screen at all. And it was a really hard time. And so I really wanted a way to engage with the students. And so one day in zoology class, I asked, does anyone want to start a reading group where we read papers about coronaviruses? And a few of them said yes. And so Team Cove Hunter was born. The funding for this project came from a combination of the Field Museum. So they did a call for innovative uses of the collection, and Adam proposed screening the tissue collection. And then IIE, the Institute for International Education, sent out a call to Fulbright alum asking anyone, if they're working on coronaviruses, to submit proposals. And I was working with these students from the south side of Chicago, which were really impacted during the pandemic. And I wanted a way to provide funding for them and to engage with them in real ways, in real positive ways. And so that's where it kind of all started. At the same time, papers were coming out making predictions about which mammalian host to screen. And so the paper on the left looked at the ACE2 receptor across vertebrates, including 250 mammals. And the paper on the right was doing statistical modeling. specifically with ecological data to make predictions about which mammalian hosts would be good to target for SARS-CoV-2. And so we wanted to use these papers to kind of narrow down which of the field museum's tissue collections to screen, but also to help inform the models, right? So that we could test the models and iterate, provide them information on any new positives. And so the team included field museum collectors, so some of the recent collectors of mammalian tissues, and including some of the recent trips that me and Adam have gone on to Angola and Democratic Republic of Congo. It also included this modeling group. So Dan Becker and some of the folks at Verity were helping to provide us lists of bats for which we could focus our sampling efforts. And then the lab folks included me and a team of students from Chicago State University, as well as Hendrik Pointer at McMaster University, who was helping provide molecular guidance, but also for any of the samples that we get positives for, sequencing the whole genome of those coronaviruses. And so the Field Museum has a rich collection of tissue samples. They have over 80,000 mammal tissue samples, and these include 18,647 bat samples. But I should say, while these are in the cryo facility, they weren't, for some of them, they weren't always cryopreserved. So not all the tissues were flash frozen immediately. And some of them are preserved in different types of buffers. So DMSO, ethanol, for example. And I think this is one of the reasons we were excited about the project and why Adam was really excited about it, is this project allowed him to dive pretty deep into this collection and see what the limitations were and really look at the tissues and see what were in these vials. And so what we learned is that the preservation method for these tissue samples were not always recorded. So the modelers came back with 423 bat species that were predicted as suspected hosts of beta coronaviruses. And when we looked at the Field Museum's collection, we found that they had 84 species that were flash frozen tissue samples that we could work with. So we selected 1,500 samples to screen. And you can see in the pie chart the number of screened samples by family. A lot of them were, the number is just the number of tissues that we screened for each of these families. You can see pteropodid bats, rhinolophid bats, and vespertilionids were a bulk of the samples. And then when you look at the number screened by family, it's the number of species screened by family. You can see that the species is reflected the same way, where it's mostly pteropodid, rhinolophid, and vespertilionid bats. You can also see the number of screened samples by year, and so that's definitely reflected in the collectors and where they were collecting and what years, and probably what years they had funding to collect. And our tissue samples spanned 11 countries. And this is also, you can see, reflects where, the curators have focused their research. You see Southeast Asia, the Philippines, where Larry's built a career working with mammals, had a large number of samples. And then in the southern sub-region of Africa, there's a good group of us at the Fields Museum that are collecting samples. And so I feel like we really knocked it out of the park with the CSU student participation in both the collections and the lab. And so the Field Museum was really gracious about letting us come into the space at a time when they still weren't allowing staff to come into the space. So they gave the students badges, and we were, in the cryo facility, and we were in the Pritzker DNA lab doing stuff. And it really, you can't imagine what a unique opportunity that was. And in terms of building their confidence, and it was great. And so, part of the challenge for me was I was trying to build this student-friendly PCR assay, and some of these papers, there are a lot of papers out there where they're PCR-based approaches, either qPCR or semi-nested PCR, that advertise that they're universal primers, or they can screen alpha, beta, gamma coronaviruses, but really that just doesn't exist. So I spent a great deal of time with the students optimizing, and we did settle on a PCR-based approach because we could convert the RNA to cDNA using RT-PCR, and then make it something that was more manageable for us to work with. And teaching students how to do stuff with agarose gels is really straightforward as well. The only problem is, for most of these, you do end up with secondary amplification. And I spent a great deal of time band cutting. And so we basically cut anything that was near the range of the fragment size we were after, and then we would Sanger sequence that. And so early on, when we found some positive individuals from our Angola samples, one of the students led a tropism test where she took different tissue types and looked at the number of viral copies using qPCR. And it's probably not surprising that the highest number of viral copies were in the back colon samples. Unfortunately, most of the samples that we were working with were not back colon samples. Other students had success in this project as well. Alasia Harshaw found alpha coronavirus in a novel bat species. So the alpha coronaviruses are shown in blue, the betas are shown in green. Corval Russell found a beta coronavirus that falls out within the sub-Saharan African clade that is closely related to SARS-CoV-2. So while the student successes were great, overall, screening 1,500 samples, we only found 12 positive samples for coronaviruses. So it was a lot of effort for very few samples being positive. And so if we look at the model classifications and the bat species that we tested, so you can see that there were 74 bat species tested that were suspected hosts, but we only found one positive. There were 21 bat species that were true positives, and we only found 10 individuals. And then there were 3 that were not in the model, probably likely due to the taxonomy issues that Kendra was talking about, and we only found 1 of those. So this leads to the question, why so few positives? I Sanger sequenced all of the bands that were in that 400 base pair region of the target amplicon sequence, and the sequences were mostly RNA, and so when I would blast them, they would mostly blast to some mRNA transcript, which indicated to me that the RNA method of extraction that we were using was working. However, like Verity was saying, tissue type might be an issue. And so the only positives that we got, those 12 positives, were from colon samples that me and Adam collected recently on our trips to Angola and DRC that were flash frozen or preserved in DNA RNA shield. And so we think perhaps the tissue type or tissue quality were factors in this study. It could be methodological though. We could be getting a lot of false negatives just based on this PCR amplicon method. And so the next step is we're sending off all the RNA plates to be sequenced. But still, this problem of not knowing, until we were really down in the cryo facility, what the tissues were. So sometimes we didn't even know, if we'd open a tube, if it was going to be muscle tissue or some kind of organ tissue, and what type of buffer it was in doesn't seem to be uncommon. Adam worked with Colleen Cronin, a master's student in conservation medicine at Tufts University, along with Dan Becker. And she was interested in using GBIF to look at both rodent and bats to see if there was flash frozen tissue available across collections. And she had similar issues where that type of data just was not available. And so the questions I have for the group are very similar to Verity's, where like from the user side, and when we're looking at these collections and looking at putting in a request for tissue, there really isn't that sort of metadata available to us. Sometimes it just says tissue, yes or no. And so knowing the history of, has it always been flash frozen? Or what type of buffer it was stored in would be hugely important. And then from Adam's side of it, as a collections person, the space, the time, the money for managing these sorts of collections. And with that, I'll take any questions.
Time for quick questions.
Yeah, the tropism of why colon? Well, I'm just curious, like, why do you, is there a reason, physiologically that it's not causing a robust infection and therefore it's getting removed through the colon? Is that where we should be looking? Because colon just seems odd to me, actually.
Yeah, the idea here is this is where they're shedding it.
Okay.
And so it kind of wasn't surprising to us that this is where we would get and I think this has been documented before for coronavirus, that feces and colon might be the way to go for screening.
Cool.
Just to add to that, since my primary work is on coronaviruses, you're correct. Feces, actually rectal swabs we found to be quite useful. Colon would be great. The issue with feces is sometimes it dries in a hard pellet. No matter how many beads you put in there during your extraction, it just doesn't break up. So I think you're on the right path for tissue types. I do find it very interesting that typically you'll see about 10% prevalence.
I think me and Adam are with our collection.
Yeah. And then also kind of back to the modeling work that was done. They have the saying that all models are wrong, some are just useful. And so I think a lot of taxonomic discrepancies sneak into those databases. And what they need to do is kind of pair up with broader expertise beyond modelers and actually work directly with virologists, disease ecologists, bat taxonomists, and that was more of a statement than a question, I guess. But
yeah, I agree.
Well, that's true. You guys do know a little something, but you were working with what you had, and I understand that like different buffers and cold chain can be a challenge, but I think technology is also advancing to the point where we're seeing whole genomes being pulled from things in RNA later, where we initially were told that's not a possibility, it's not going to happen, and then they overcome obstacles with new technologies, just keep trying kind of type thing.
The next talk, actually, this targeted enrichment approach. So say you just have a few reads of a viral copy, using this bait approach is really useful. So we are working with Henrik to do this on some of the formalin fixed stuff.
Great.
Thank you, Molly.
Thank you.
So again, if there's additional questions, the Google chat does, or Google Sheet or Doc or whatever has something that you can communicate in. Okay, our next presenter is stuck in Phoenix, and is Neal Platt, who's stuck in Phoenix, there he is. So Neal is at the Texas Biomedical Research Institute. As Pam tried to mention earlier, he was trying to fly here, but from San Antonio, not Charlotte. Yeah, and I've noticed the weather. There was some bad weather in the DFW area, and they put him in Phoenix, and he's been in Phoenix for 2 days trying to get to Kansas City. And he can't get back to San Antonio at this point. So he's going to give us a presentation from the airport. So Neal, I don't know if you could hear us okay. If not, if you can, go ahead and go ahead and start.
Can y'all hear me?
Yeah, Neal, you're good.
You can hear me? Okay, great. So today I'm going to be talking about some work that we started in our lab just right before the pandemic, where we were trying to use museum specimens to extract historical information on a helminth pathogen that we study. And my slides won't go to the next slide. So I don't need to emphasize with y'all. We all know that the museums that we have an extraordinary amount of pathogen information kind of housed there, but we need to be able to figure out ways to extract that information. So there are a lot of different tools that we have to be able to do that, whether it's just kind of shotgun metagenomic sequencing or Amazon sequencing and so on. Unfortunately, there's no perfect set of tools, and they all kind of have their positives and negatives. So for example, like a host depletion strategy where you take a sample and you try and remove all of the host DNA and only sequence the pathogen is really useful, but a lot of those methods are host specific. So the methods that you would use to be able to, for example, deplete a particular bat species might not work for rodents, or might not even necessarily work for other closely related species. In an ideal world, though, we'd be able to have tools that are kind of tissue agnostic. They have kind of minimal taxonomic restrictions. They're high throughput. We're able to generate a lot of data cheaply and quickly. And things like they're, repeatable, and we can also kind of maximize the amount of genetic information that these methods provide. I don't know why my slides aren't going, so I'm just going to do this. So one of the ways that we chose to do this is to actually use a pathogen enrichment method. And to do this, we took a modified UCE-based protocol. And in this protocol, I'm sure a lot of people are familiar with it. So it targets highly conserved regions in genomes of a wide range of species. And this is a really powerful method, and what you end up with are probes that are effectively capable of capturing homologous sequences in species that are hundreds of millions of years diverged. As you can see, there's an example here on the right side of my screen where probes were designed to identify these loci in all hymenopterans. One of the cool things, though, is because these probes are sticky, is we can design probes for taxa A, but then we're able to actually use those same probes to get information from taxa that we may not have data from or don't even know exists, because these probes are capable of capturing sequences up to 20% diverged. So what we did is we took this UCE protocol, and rather than just targeting one specific group, we decided to grab a bunch of different pathogen groups and kind of develop independent probe sets for those pathogen groups, and then combine them into one potential assay. So kind of the end result of this is we have a probe set of 40,000 loci, where each of the, or 40,000 baits, where each of these baits are used to target particular loci in up to 37 different pathogen groups. And each of these pathogen groups are captured by at least 50 different conserved loci. So everyone is going to be interested in the pathogens. Here is a quick kind of table of the pathogens that we particularly targeted. So the thing to keep in mind is the way this probe set works is while these are the pathogens that we targeted, our probes that we design will catch all rickettsia, not just the rickettsia that are shown here, and should theoretically catch all helminths, not just the pathogenic helminths shown here. So we tried to make it as flexible as possible. So the first thing that we did after we developed this toolkit was we actually kind of tested it on some lab controls. We took some lab mice and spiked it with some pathogen DNA that we had floating around in the lab. And one of the things that you'll see is that with a couple rounds of enrichment, or with one or single round of enrichment, we're able to start driving up the coverage of these homologous UCE loci, even in samples that were spiked with only 0.1% of these particular pathogens. More importantly, though, is not only are we getting the reads from those individuals, but those reads aren't randomly distributed. And because they're from those particular loci that we're interested in, we can use that to actually place those pathogens in phylogenetic trees that you'll see here on the right side of the screen. And for each of the controls that we examined, we were able to actually put them into the tree next to the appropriate sister taxa. So this was a good kind of control study and kind of proof of concept. So we actually tried it in some real samples, and we're interested in schistosomes in our lab, and these are parasitic helminths in Africa. So we grabbed a couple samples from the Texas Tech Museum that were collected by Adam Ferguson and Molly McDonough, and we looked at liver tissue, and we tried to see if we could actually pull out potential schistosome parasites from these samples. So the end result of those analyses you'll see here is a heat map, and there's a couple things that I want to point out. First is you get like some kind of low intensity hits to a lot of different types of species, but you'll see that Plasmodium is present in almost every sample that we examine. This is a bioinformatic artifact and something that we should talk about in the future. But you'll also see that nearly every sample we had some level of Bartonella. In particular, some of these samples were up to 50% of the data that we sequenced were from these different species, Bartonella species. Now, as I mentioned, we're able to take that information and place it within a phylogenetic tree. So when we did that, what we found was that the species that we were recovering fell kind of in various places through the tree. So we weren't capturing a single particular species. Some of the things that we found we were pretty excited about. You'll see at the very top of this tree, Bartonella mastomitis is a species of Bartonella that was described in Mastomes from 2018. However, we're actually finding these sequences and samples that were collected sometimes 10 years prior to that. So this is an example of a bacterial, pathogen, commensal, however that natural history is with those hosts, but it existed in a museum specimen sitting, waiting for someone to be able to actually go out and pull that type of information. So there are other things that we could potentially do with these tools, like being able to extract population genetic information and get an idea of like the effective population size, or we can go back to these samples and kind of retarget them now that we know that they have a particular bacteria species of interest, and we could sequence whole genomes of those particular species. You see this type of technique done frequently with things like the plague. So I have a lot of kind of concluding thoughts, but I think the big thing that I wanted to point out is that, our goal was to try and develop like a tool that is fairly agnostic. And in that process, we had a lot of obstacles, and there are a lot of things that we still need to work on. Things like our enrichment is not nearly as efficient as we would like, and we would love to be able to get that up to where, rather than 10% of the sample being a particular non-host DNA, get up to higher than 90%. There's lots of problems with bioinformatic false positives. So all the Plasmodium samples that we found that weren't in our controls were primarily due to the fact that mammal genomes have AT repetitive rich regions and the plasmodium genome is extremely AT rich. So you pick up these kind of false positive hits. The other issue that we have is funding. So we have approached NIH just to see kind of baseline interest in something like this. And it's so out there for them to be working on this type of stuff from museum specimens that they don't even know how to handle this. So the next obviously available opportunity is NSF, but our institute isn't able to work with NSF due to overhead restrictions. So the other thing to point out is there are a lot of potential kind of alternatives to this type of method that may be useful. I think this isn't going to be a one-size-fits-all option, but the tools are being developed to potentially be able to like scan, or not scan, but take a single tube and assay for a large number of pathogens in a single run. So that I have lots of people to thank. In particular, Elisha, shown here, he is a postdoc in our lab who really helped me a lot with developing the wet lab protocols. I'm a computer guy, so I sit at the desk, and he does all the black magic hard stuff in the background. And obviously, we had a lot of work from people at various museums who not only collected but also archived those tissues. So with that, I'm going to stop sharing the screen, and I'll take any questions.
Okay, Neal, I think we can do questions. Is that right? To the microphone? I don't know if you can hear me okay or not, but. I think everybody was oohed and awed that you were able to present in the Phoenix airport without too much distraction. We have a question from Rick White.
Gotta have a question for a fellow computer guy. I mean, come on. All right. So Neal, taxonomy is hard, right? Like it's just hard to get it to where you get into these genomes, and we struggle with this a lot, especially if we were trying to find something like a retrovirus in a sample, because there's all these repetitive endogenous EVEs in a genome. Like, just as a fellow computer guy, what are your thoughts on trying to help with the taxonomy of these things and like to kind of fish out some of these things? We've been looking at CNNs and trying to remove the host stuff with like deep learning stuff, but I'd like to hear your opinion on this.
So I think our goal, I guess, is twofold. Is one, to be able to remove as much host in the tube as possible. So, if we can do it that way, that would save us a lot of problems. But on the back end of the taxonomy, luckily for me, I've not been too, I'm not going to ever be an expert in the taxonomy of any of these organisms anymore. But if we can get those spit samples into a tree, then it allows the people who are interested in those, like we were looking for schistosomes. We found Bartonella. I didn't know Bartonella was even a thing until we did this study. So it kind of takes the onus off of us and allows us to get the information into the hands of the people who are the most interested and most best situated to handle that.
Okay. Neal, thank you very much, and thanks for making it work. And Neal, when you get a chance, take a look at the Google Doc for additional questions. Okay. So for our last presentation for this case study portion, before we move on to our discussion, have Rick White. Oh. Okay. Sorry, I had a little bit of feedback there. Okay, so Rick is coming to us from UNC Charlotte. He'll be talking to us about preventing the next viral pandemic via elucidation of mammalian collections. So Rick?
Well, again, thank you all for the trip, and again, Pam, Deb, and I'm just really excited to be here. So I'm a computational molecular virologist. I'm at the University of North Carolina, Charlotte. I'm involved in our new Cypher Center, which is a $10 million retrofit to kind of prevent the next pandemic. And so today, I'm just going to go through more of a physical, kind of a philosophy kind of talk. Not a lot of data today, but I'm here to discuss. So here we are, folks. Here we are. 6 million people dead and over 1 million dead Americans from COVID-19. One virus, a zoonotic virus, emerged from a pool of 10 to the 31 viruses. So that is our current estimate of viruses on the planet. That's 10 orders of magnitude of the stars in the observable universe, right? So there's a potential pandemic everywhere, right? And the question I get almost daily is, how do we stop the next one? And so the answer here, of course, this got cut off, is animal surveillance, period, right? And so unfortunately, every disaster movie starts with warning with limited action. So I want to give you a paper that comes to mind when I saw this kind of creeping up and seeing in. And so you haven't seen it, here it is for today. So there was a group in Hong Kong that talked about surveying acute respiratory SARS-like viruses. And they made this claim in their paper back in, and you can see this in October of 2007. The presence of a large SARS-CoV-2-like reservoir in horseshoe bats, together with a culture of eating exotic mammals in southern China, is a time bomb. The possibility of a reemergence of SARS or another novel virus, therefore, should not be ignored. Here we are, folks. All right, I wanted to also talk about a couple of my some other thoughts about zoonoses and reservoirs. So all animal viruses, and all viruses for that matter, start in something as simple as a bacteria. And they evolve through trophic scales. They move through vector species such as ticks or mosquitoes or other mammals to us. There's a huge spillover rate from animal to human. This happens daily, all the time. And then eventually you have one that goes from human to human. All pandemics arise from animal reservoirs. So to talk about coronaviruses, to what Cody was talking about, from the best of our knowledge, most, if not all, have a bat origin, including HKU1. This is a paper from my friend of mine at Cypher, Dennis Jacob Machado. We have not found some of these other bats that represented these other lineages, but these are the recent spillovers from intermediate hosts, potentially. Bats provide a huge and extraordinary reservoir with viruses of pandemic potential. And this is an older paper from 2020, but as we surveyed for, over 3,700 new coronaviruses in bats alone. Also paramyxoviruses that we'll talk about in a minute. These are measles-like viruses, and then rhabdoviruses, and a variety of things that we don't really know. And so these are up-and-coming viruses waiting to become the next pandemic. So I want to give you a history lesson on cautionary tale. It was great. We had a pox virus expert with Molly. It was great talking with Molly. So this is a shout out to smallpox, unfortunately. So I want to talk about smallpox and a paper that Dennis Jacob Machado and I wrote. So smallpox, in the last 100 years of its existence, killed 300 million people. It is second to only Yersinia pestis in the amount of human casualties, and it's potentially now tuberculosis is actually, I think, surpassed Yersinia pestis. So if you took a novel pathogen that had the mortality of smallpox, of varroa major, which killed around, on average, 3 out of 10 people, and it infected 50% of the U.S. population, within the same time frame of COVID-19, you would look at total U.S. societal collapse and over 50 million dead Americans in the same time frame. So the question I also get asked is, how do we measure this? And it was brilliant talking to you all today and how we can work together to figure this out. The majority of viruses aren't frozen, and samples are not frozen. The majority of viruses pandemic potential are RNA, and we all know that we've evolved as eukaryotes to make RNase and remove them, RNAi and stuff like this, and so RNA is not very stable. DNA obviously usually work, but again, formalin is problematic. And then while RNA, DNA may be present, they may not be intact. They may be fragmented and these kind of things as well. So the question, the next ask, and this again came from a conversation with Molly, what can we do? How can we figure this out? So I offer a new way, protein-based. Protein inherently might be more stable. And so I and my lab group, and of course with the grant that Cody and I got together with the NIH, are starting to build antibodies for bats relating to the immune system. But we can also build these for viruses and novel viruses, and we need pan antibodies to scan potential new pathogens. The other one is mass spec, or tying them two together, maybe even doing on a targeted enrichment, which also Neal talked about. But that's the current technology, right? But just around the corner, folks, there's a new technology that we can have in the palm of our hand that will be able to sequence proteins directly. There's been 2 papers that have been recently pushed out, and so eventually we will be able to sequence full proteins, full proteomes out of these samples. And this is just only a matter of time to where we're able to sequence and look at the proteins, right? Again, it's a different sample prep, it's a different type of preservation, but I think we should work together and we should test to see if this is possible, because when we can sequence proteins, we know the organism is there, even in the absence of RNA or DNA. My lab group, again, I'm a computational biologist, so I want to tell you about some computational tools that my lab group has put together that I hope all of you can take a look at. And if you have problems, let us know. So we have built this infrastructure called the Titan infrastructure. It is a DNA to lipid analysis, whole multi-omic, meta-omics, any kind of data you want to put together. This program will help you going from start to finish, from raw data to knowledge. As well, if you're out in the field and you're trying to figure out what samples are different and you have a nanopore device, you can use this thing called Meerkat, which allows you to rapidly tell whether samples are different using a camera counter. And then lastly, that's part of the Titan infrastructure, both these other things, Metacerebrus, which is the first universal annotation tool through the entire tree of life. So you want to know the functions or what's going on in your virus, your bacteria, even your bat. This program will functionally annotate your genome on your laptop. So how am I doing on time? Well, I'm doing pretty good. All right, fun. I get to talk about the top 3 viral suspects for the next pandemic. Fantastic. I got 3 minutes. All right, so who y'all think is going to be number 3? Well, I'll give you one right here. It's called Henipavirus, and these are paramyxoviruses. They're in paropid bats, and they have extremely high mortality. Henipa and Hendra, obviously. The last outbreak actually was in 2022. This is a little bit older slide. They have a very, very high mortality rate. And these are measles-like viruses. We don't really understand how the virus transmits very well. We assume that we put them in BSL-4 containment. We assume they transfer through respiratory tracts, through droplets. But all the folks that we have done this last outbreak in Carroll, I think in 2023, it looks like tree sap could have been the reason this virus passed on and jumped from bat to human. But again, this is a virus of a virus that has a high mortality with high respiratory transmission, is a virus we need to keep our eyes on. I'm going to give you 2 that may have not really heard about or maybe even thought about, but I want to have you keep it on your radar. And that is the arenaviruses. So the arenaviruses affect everything from reptiles, fish, bats, you name it. They are negative sense strand RNA viruses. The Lassa virus is the most common. In Lassa viruses in West Africa, around 80% of the cases are asymptomatic with a 1% mortality, but a third, even those asymptomatic problems, lead to deafness. So it doesn't necessarily have to kill you to cripple an economy or to hurt humanity. Again, they have massive taxonomic ability across everything from reptiles, from fish to humans, and they jump in and out all the time. Again, of course, I have to give you my dishonorable mentions of viruses that could potentially be pathogens and cause pandemics. One of them is obviously the Caribbean Congo virus, which is a tick-borne encephalitis virus. We have found potential cases of it going from human to human. Marburg and Ebola, obviously in Rosetta's bats, the MERS and camels, monkeypox and potentially Alaska pox or boreal pox. There's a large niche from the eradication of smallpox for a pox virus to fill the niche and become a human pathogen again. Influenza A, obviously, with bird flu, it's getting found in milk and tissues. It's something we have to keep our eyes on. And then lastly, what the WHO calls virus X, a virus of unknown origin or unknown family that causes a global pandemic. But my number one suspect, which I put a lot of time in, and Cody and I have gone back and forth, and I've got 30 seconds maybe, is hantavirus, and I'd love to talk to you about that. And then lastly, how do we prevent it? We need a better relationship with animals. And then RNA viruses that causes a respiratory tract infection can evolve into something that we've never seen before and cause a pandemic. And I want to thank the Cypher team and, of course, my amazing grad students and collaborators and Dan Janis and Cody Thompson for all of this. And so again, we're highly collaborative. We're here to help. We're here to give potential services for sequencing, proteomics, you name it, computational support, and let's prevent the next one together. Thank you for your time.
Any questions for Rick?
Yeah, Kendra.
I'm interested to understand your reasoning for expecting viruses that have high mortality rates to actually develop from outbreaks, which is what we've typically seen, to pandemics, because they typically burn out rather quickly, and that's why they're usually limited to outbreaks.
I just don't want it to happen.
Agreed, but
just because it hasn't happened doesn't mean it won't.
Yeah.
And it doesn't necessarily have to have an 80% mortality rate if it had 10, 30%, but smallpox had a high mortality rate at 30%. And it was, obviously it's kind of mixed because we also had Virome minor, which had 3% mortality, and we can never kind of decouple the two of those, and it's unclear whether minor was more globally distributed versus major. This is something I'd love to answer. But it's not necessarily the mortality rate. It's just something from those families could emerge that we could be completely blindsided by. Grab a picture of me too. Way to think I made it up.
Thanks. I had a question on the nanopore slide. Why use the proteins? Why were you trying to sequence the proteins specifically? Is it just to get at the low abundances in samples later, or like a field-based approach?
So the problem with RNA is its labeling, right? And if we're trying to find new viruses, in having cold chain and stuff like this, if we could sequence the protein, we don't have to worry about all this worrying about whether the sample's absolutely cold or these type of things. So it gives us a strategic advantage in the field finding viruses. So I think protein is the way of the future.
I have what might be an outlier question.
Sure.
In part of this between Viral Muse and the working group with CTAF and the conversations that we had in those meetings, one of the things Quentin Groom pointed out, and then there was a paper, was looking at something like iNaturalist and the collection of data by humans to share that basically said, we don't want your pet data. And yet we have evidence, and you guys know more about it than I do, to say, you can get a cold from your pets, or they can give something to you, or you can give something to them. So are we, or do we need to also be thinking about that and how we engage people in getting more information about that, about transfer between humans and their interactions with the potential carriers of these things, or us giving it to them?
Yeah, no, this is a great question. So going back to Cody's slide really quickly. So there was an outbreak of SARS-CoV-2 in mink farms. And it was very clear that it wasn't the minks giving it to people, it was us giving it to them, and then the minks giving it back. And so Dan Janis's work at Charlotte showed this when he was at Ohio State, that it wasn't necessary for this mixed signals, right? And it's pretty clear. And even the one with SARS-1, where you had the civet, you could never tell whether it was the man who gave it to the civet farmer, or the civet gave it to the man, right? So, and we definitely do see in the case of, even I think the president's dogs got SARS-CoV-2 from Secret Service types of folks. So I think surveilling even our domestic animals, I think, is also worth it. The benefit of wastewater is we kind of get a snapshot of this. We don't get the carriage of the linkage, which is kind of what Kendra is talking about. I think that's really, really important to have that linkage. But again, as sequencing gets cheaper, it's better. But again, we're always stuck because RNA is super unlabile, super labile. Now you can directly sequence RNA now on nanopore, but even then, it's just compared to protein. We want to go back and really understand, going back to the 60s, going back 100 years, and keeping these collections in the long term. I think protein is the way to go, and much less energy consumption for protein than -80s in liquid nitrogen.
Okay, I think we'll pause there. Thank you to all of our speakers. So we're going to take a quick 5-minute break to allow everybody to go to the restroom, get coffee, water, and we'll reconvene with Deb leading the charge.